everyone. Happy Tuesday. Thanks for joining our AP Biology live stream tonight. My name is Kari Parnan. And all right, tonight it's going to be starting off with actually a free response from last time. So if you were not a part of my stream on Sunday, it's no problem. But we focused on the structure and function of biological macromolecules, most specifically nucleic acid and DNA. So we'll do that. We'll finish up the free response, and then we will start Nucleic Acid 101 and looking at those three main things there, okay? So last time you know what? Time out. I'm going to first tell you, help you understand the language of the AP College Board as well. I don't know if your teacher does this or not, but there's these things called enduring understandings and then objectives of how we're going to get there. So the big understanding for today and for all of this unit is that heritable information provides for the continuity of life. And that sounds very teachery, and you're like, what the heck is that? But it's just laying the groundwork for what we're learning now. It has to do with information being passed down from one generation to the next. And continuity of life has to do with how in common this is among all living things. So continuity is a weird word in biology because it makes you think of maybe the word continuous, but the idea is we all have this similar or same framework of genetics, and that provides for similarities among all living things. But in a little better language that's more maybe student-friendly, I really just by the end of today want you to be able to describe structural similarities and differences between DNA and RNA. And that might sound really easy. I always have students who are falsely confident about DNA and RNA, and they say, okay, yeah, I get it. Like DNA has these bases, DNA is double-stranded, yada, yada, yada. I know the difference, I'm good. But then they get a question that really gets to whether or not can they apply those differences and make a decision. They kind of trip themselves up. And so just make sure you know this stuff very well. So today, ideally by the end, you should be able to compare and contrast the nucleotides that make up DNA and RNA. Think critically about why DNA and RNA have differences, and then also start to ask why when revealing bond types found within these nucleic acids. So if you've been a part of my streams before, I have talked about how in AP Biology, we want to move you beyond just memorizing things and saying, I know everything there is to know about DNA and RNA. That's great. And sure, that's great. You should know everything there is to know. However, but have you thought critically about it? Have you asked why? Instead of just memorizing that this is a covalent bond and this is a hydrogen bond. If you're able to really understand why that matters for life, then you're going to be in a much better position of understanding this material on a higher level. And so you'll get to those free response questions on the AP test, and you'll be able to explain yourself and not just spit out textbook information. You'll be able to be a really, really good explainer. So here's a quick review. Here's another thing I want to also give you some practice in. The types of questions for multiple choice that you will see on the AP test, the questions are several sentences long, like this one, okay? So the question stem is, we have a purified protein resuspended in water was examined. Scientists found that amino acids with non-polar R groups were buried in the middle of the protein, whereas amino acids with polar R groups were found on the outside. It's asking which explanation below most accurately describes the location of the amino acids in the folded protein. So in your chat to me and everyone else, jot down how would you, or what makes something a polar R group? So go ahead and just type it in. If I call something a polar R group as a part of an amino acid, what might be true about it? Is it uncharged? Is it charged? Think about that. So go ahead and type in an answer just to think about what does a polar R group mean? So polar R groups, what should be true about them? Remember, think back, water is polar. So if something is a polar R group, is it going to like water? Oh, there we go. Dakota. The electronegativities are different or unequal. Yeah? Okay, good. So they're going to have some charges, right? So some partial charges or some full-on charges. Good. So if you're polar, you could have some unequal charges. Absolutely. Are they going to be hydrophobic or hydrophilic? Let's get to the answer options here. So this is a multiple choice question. I just wanted to give you guys the STEM first. So which explanation most accurately describes the location of the amino acids in the folded protein? So A, polar R groups on the surface can form ionic bonds with charged ends on the water molecules. Polar R groups are too bulky to fit in the middle of the protein and are pushed to the surface. Or C, nonpolar R groups that cannot form hydrogen bonds with water are pushed into the middle. And D, nonpolar R groups from different parts of the protein form covalent bonds with each other to maintain the structure. So I know these are wordy, but like I said, I'm trying to give you some experience looking at these more wordy questions because this is what the AP test does look like. Okay, so in the typing box, chat box off to the right, go ahead and answer A, B, C, or D. So nonpolar groups are in the middle, polar groups are on the outside. Which explanation makes the most sense to actually explain why that happens? So you can just type in A, B, C, or D, and then we'll go over it. Awesome. All right, so everyone is saying C, absolutely. So the non-polar, right? We know that if you're non-polar, you're not going to want to interact with anything that is polar, right? If you're non-polar, you don't go with anything that's polar. So they're not going to be able to form hydrogen bonds and they push to the middle. And that is just one aspect of protein folding. If you were not a part of our last stream on Sunday, I would recommend going back because we really went into our groups and their behavior and why they are the way they are. Okay, it looks like we've got a few new people. So thanks for joining us. We're just doing a quick review from the last stream. Okay, so I posed a free response question at the end of my last stream. We ran out of time, so I kind of gave it as some homework. If you were not a part of it, don't worry about it. You can just still look at it with fresh eyes and think about what you would say. So the free response prompt says, the physical form of cells and organisms is influenced by structural polymers. Part A, for each below, describe the structure and role of each in an organism. And B, predict the cause and or effect of a disruption in one of these polymers. So the two things it's wanting you to look at are DNA and cellulose. So the AP biology exam will give you lots of part A, part B problems. I like to solve them or explain them individually. So some students kind of like to bounce back and forth between A and B, but if I'm the person reading your free response answer, the more organized you can be about how you answer it, the easier it's going to be for them to score it. Because remember, you're being scored by a human. So humans are scoring this, so make it easier on them, okay? So in the very beginning, I know this is kind of all the way already filled in, but part A asks you to talk about structure and role. So obviously, in a free response question, you would write this in sentence form. I wrote this out more like a rubric. Okay, so for DNA, you can give yourself 1 point for mentioning something about the structure. So the fact that it's made up of nucleotides that are the monomers. Or you could have talked about how it's a polymer that's a chain of nucleotides, which is basically saying the same thing. And so that's what I like about the AP exam is that you can get points for not just saying one thing, right? So like if you make it clear you know DNA structure, they will give you credit for it. It doesn't have to be word for word what someone else does. Okay, so that's the structure of DNA. Role. What's the role of DNA? What's its job? Genetic code or carrier of genetic information. So if you would have said either of those, you would get a point each. And then cellulose is a carbohydrate. So mentioning something about its structure, that it's a polymer of glucose molecules, and glucose is the monomer, and that its role is to structurally support plant cell walls. Okay, so be really methodical with your answering and always go back to the prompt and say, okay, did I talk about structure? Yes. Did I talk about role? Yes. Moving on. Okay, so self-check that way. Part B wanted to look at predicting the cause and or effect of a disruption. Okay, so you have to start thinking about, would we have to mention each of these bullets as long as we have one? Yep, you could just answer one of those. So someone just asked if we went back to the last slide, would you have had to answer all of those things, right? Like say, call it a nucleic acid, talk about the monomer, talk about the polymer. You wouldn't not need to hit every single thing. Those are examples of if you talked about one of those, you'd be fine. And the more details, the better, obviously. So if you were just calling it a nucleic acid, you might want to define what a nucleic acid is. And in your definition of that, you'd probably stumble into talking about nucleotides, right? So yeah, those are usually, if you look at previous free response rubrics for the AP biology free response, it's really open in terms of how you can get your points. There's a lot of things you can mention. Again, they're not looking for just one specific answer a lot of the times. There's usually like 5 examples you can choose from. It's really nice, which is what I actually really like about it. Good question. So part B, I'm going to start with DNA and talk about possible causes and effects. So here, same thing. The first 2 bullet points, as long as you had mentioned something along those lines that, okay, well, DNA altered would probably be from a mutation, right? So that's changing a base. That'd be your cause. If you wanted to talk about the effect, that'd be the second bullet point. And so if the sequence is altered, when proteins are made, it's going to make the incorrect protein. Usually, I didn't write this in the free response question, but usually with a question like this where they're wanting you to describe, structure, functions, or mistakes, there's usually some sort of explain the significance of, right? And so really explain, what does that all mean? So imagine you finish writing your free response question, you talk about mutation, you talk about why that would mess up the proteins and how that would affect a cell, because proteins have jobs, right? So if a protein can't do its job, the cell is going to suffer, okay? So always connecting it back to, why does this matter? What's the big picture? So I don't know, I would always tell my students at the end of a free response, if you haven't made it clear, like why what you said matters, then maybe go ahead and explain why that actually matters. Okay, so that's for DNA. If there was a disruption in protein, there's 2 reasons why that could happen. The first bullet point takes care of that. If there's incorrect or disruption of the protein, that could be caused by DNA, number 1, right? So if DNA is mutated, then the amino acid's not going to be the correct amino acid sequence. But the other thing that can affect or disrupt a protein is just the change of environment. You can have a perfect protein, but if the environment changes and all of a sudden the pH levels drop or rise, or the temperature drops or rises, things like that, you can have unraveling, you can have denaturing, or you can have permanent degradation of the protein itself. So those are 2 avenues. And again, if you mention one of them, you wouldn't have to mention the other. And then also finally, like I said before, explain the significance of chemical interactions. So really go in and show that AP reader you know what you're talking about. Don't just say, oh, the protein would be malfunctioned, right? Because it's like, all right, well, why? Explain why. Explain, well, proteins have a function because of their shape. Why do they have the shape they have? Because they fold in a very particular way, right? Okay, so make sure you just really, really, really explain that well. And we'll just keep getting more practice with this kind of stuff. All right, next. So on to DNA and nucleic acids. We know the nucleic acids are DNA and RNA, right? So I just drew these with marker. And things that we know already, it's the genetic molecule, right? These are things we know. Hopefully before you logged in today, this is stuff you knew. Okay, so it's the genetic molecule. All living things have it. It's the language of DNA, and the language is from 4 unique nitrogenous bases. Okay, so I know some biology teachers use the analogy, like our English language comes from an assortment, an organized assortment and sequence of 26 letters, right? We have 26 letters in the alphabet, and then we have this huge language that different words are put together. They're different sequences of words. In DNA, it's incredible that there's only 4 letters that we're dealing with. So 4 unique bases and their actual sequence and how they're organized, whether it's 10 T's in a row followed by GC, GC, or it's unbelievable how 4 bases can create such diversity for all living things. So that's something hopefully we already knew, that it's just 4 bases that we're dealing with. And then finally, this is something you definitely should have known already, is that polymer, it's a polymer built from nucleotide monomers. And so if I asked you, what's the monomer of a nucleic acid? You should say a nucleotide, okay? So those are things you should know already. Okay, the other thing, just wanted to put this in here. If it comes up in your classes, describe the structure of DNA. If we were to just roughly describe the structure of DNA, a lot of teachers call it like a twisted ladder. So you've got your ladder here on the left. If you were to twist that around, that's what the DNA double helix is called. So I didn't want to go through an entire day of nucleic acids and not talk about the double helix. Okay, so it's a double helix. There's 2 strands for DNA at least. And it winds around like a helix, okay? If you're taking notes right now, which I hope you do, I think that it's really good to, if you're watching a screencast like this or live stream, to take notes. I think it's really helpful. So if you are, and even if you're not, go ahead and make a T-chart or a Venn diagram showing DNA, both, and RNA. So if you're more of a Venn diagram person, that's fine. I don't love the Venn diagram because the middle is so small. And sometimes you lose sight of what they have in common when you're comparing 2 things. So just a quick T-chart, DNA, RNA, and both. So our 2 nucleic acids, our DNA and RNA, and we'll be filling that T-chart out together. So that's why I wanted you to do that. So I'll kind of be clicking back and forth between new information and things like that, and then we'll go back to the T-chart. Okay? All right, so DNA versus RNA. If you were to look just at this diagram, go ahead and type in your chat, what is one thing that's different between RNA and DNA? So go, type it in. What's something different between RNA and DNA, just looking at this diagram? Okay, great. So DNA has 2 strands, RNA has 1. Thank you. Yep, Madeline, good. Good. DNA has thymine, right? We can look at the RNA over here. There is no thymine. We've got a different base. Anything else? Good. Someone said uracil is an RNA, so that's that replacement for thymine. Anything else? All right. Yeah, I would say from this diagram in particular, though, that's the information you can pull out from it. So really, really good. Okay. Let's move to the next one. Okay. If your teacher has not talked to you about this yet, no, never mind. Sorry. Strandedness. So someone said 2 strands, Madeline said 2 strands versus 1 strand. So absolutely. That's like the first thing that jumps out at everybody is that DNA has 2 strands, RNA has 1. But here's something interesting that I have a feeling this is going to come up on the AP exam where they're going to explain maybe some data about why DNA and RNA are even exist in the first place. So if you think about it, which strand most likely came first, RNA or DNA? Maybe your teacher has already talked about the RNA world hypothesis. Looks like it. Good. Yeah, so a lot of you are jumping right on board RNA. It's not anything we know for sure. It's called the RNA world hypothesis. And there's a chemist, his name is John Sutherland, and there's a really good 10-minute video that he was on with Neil deGrasse, actually, and they did a really good job explaining what he did in lab. But basically, he created conditions in which the building blocks of RNA were able to assemble themselves naturally, and it was a big aha moment in science of, okay, well, because DNA is more complex, it's got 2 strands, RNA is just 1, but they weren't sure how could it self-replicate, right? Because with DNA, it can split and replicate on each side. So that's something that might come up, thinking about why RNA might have been the first molecule, first genetic molecule at least. All right, monomer of RNA and DNA. It's a similar monomer. I wouldn't call it an identical monomer. It is a nucleotide for both. But if we're looking at the diagram right there, of course, it has the phosphate, the sugar, and the base. Phosphate, sugar, base. If you remember that really old song, not maybe really old, but you know that All About That Base song? All About That Base. I always tell my students it's phosphate, sugar, base. Phosphate, sugar, base. If you can't remember the order in terms of where they're connected. But definitely know the 3 components of a nucleotide. In this first diagram, we have deoxyribose. So if you look at the hydrogen connected to the second carbon, it's just a hydrogen, right? Deoxy means we've lost an oxygen or without oxygen. So there it is, deoxy meaning without the oxygen. So what's the big difference between the type of sugar found in RNA and DNA? So that's the deoxyribose sugar. And ribose sugar is just ribose. So looking at that diagram, okay. And more specifically, so someone answered that RNA has oxygen. That's absolutely true. Does anybody remember what's the name of that functional group that's hanging off right here? An OH. Does anybody remember the name of the functional group? Have you done functional groups in your bio class yet? We learned about carbonyls, and it is a hydroxyl group. Yeah, Ari, you got it. Good. Yeah, your hydroxyl group is your OH. So your ribose sugar has a hydroxyl group off of the third carbon where your deoxy doesn't. And so just make sure you're able to identify that as well. Okay, so first concept check of the night. Number 1, nucleotides that make up DNA and RNA are made up of identical components. A for true, B for false. Excellent. Everyone did great. Okay, thank you. Yeah, so nice job, everybody. So false, okay? This is kind of a question where it's really getting at how well, first, can you read the question of being a skeptic when you see words like identical, or anytime there's a question that has like, this is true for all, be a little skeptical about that. So the nucleotides are not identical, okay? Because first of all, just in this picture right here, they're made up of different sugars. So that middle component, phosphate, sugar, base, they're going to be different. And we also will learn in a minute all the other differences as well. So if you go to your T-chart, hopefully maybe you've been filling it in as we go. It does help to kind of keep filling it in as you understand. But we've got DNA contains deoxyribose. RNA contains the ribose sugar. DNA is double-stranded. RNA is single-stranded. They both are made up of nucleotides, but not the same identical nucleotide. Because if you look at that second bullet point, the bases differ from one nucleotide to the next. Okay, so the base differs from one to the next, which is what you guys mentioned previously when you said, you know, this one contains thymine, this one contains uracil, so on and so forth. Okay, so just to get a look at our nitrogenous bases and the differences between them. Again, a lot of you already participated about this and talked about how, hey, I'm looking at this diagram of RNA and it's got G, A, and C, but it has a U, and that's not present in DNA. So it's this component that I have boxed in that teal color. That's the base that we're talking about. That can be any of these four for RNA. So U, G, A, or C. I would recommend knowing what each of those letters stands for. So don't just say, oh, base U. Make sure you know it's uracil, right? And make sure you know like T is thymine and G is guanine, and know how to spell them too. You're not going to be taking points off on the AP exam for spelling necessarily, but you might make a silly mistake and call something like ATP when it's really adenine or something, and it's not a spelling error. That's a content error. And actually on the College Board said that at last year's free response questions, students received points off when they were talking about a protein and they kept spelling it proton, because proteins and protons are very different. And so even though everything around the word protein was right, they kept calling it a proton. And then that just made the reader think the person didn't know what they were talking about. So just be careful and know how to spell some things. All right, so here's a quick question for you. If we've got just this model that I drew here of DNA, and something you've probably already learned, right, hopefully by now, is what pairs with what, right? So G and C pair together and A and T pair together exclusively in DNA. There's no exception. It's not sometimes G pairs with A and sometimes T pairs with C. They chemically cannot and they won't. So G and C are together and A and T are together. So this is a question I like to give my students on small quizzes here and there. But if I were to give you a rough percentage of what I have for G and C, figure out what A and T are. So this is concept check number 2. Using Chargaff's rules, if you haven't learned about that yet. That's not anything that's going to be content related and on the AP exam, but a scientist basically figured out sequencing that the number of A's and T's should be the same and the number of G's and C bases should be the same. So I've given you percentage of G and C, 28% each. So find the percentage of thymine and adenine within the given DNA molecule. Oh, nice. You remember doing this? Good. Good. So ideally, the idea is your percentages should add up to 100, right? So if G and C both have 28, that adds up to 56, okay? So you only have 44% left. And so 44% needs to be split between T and A. And so that's why your answer is B. There should be 22% A's and T's. All right, great job. Let's move on. Okay, purine and pyrimidine. So this is something that maybe your teacher went over. Some years I really dig into purines and pyrimidines, and some years I don't. But I think it's because we've been spending so much time talking about chemistry, I really want to focus on at least looking at the molecules and making sure we're able to figure out why hydrogen bonding happens between the bases. So on the left, I have our purines. Okay, so they have 2 rings. Okay, so that's why they're classified as such. We've got 2 rings. So A and G are purines. And then your pyrimidines are your single ring bases. So thymine, cytosine, and uracil. And don't make the mistake of some students will think, oh, the purines pair with purine and pyrimidine pairs with pyrimidine, but that's not true, obviously, because G and C pair together. So you've got a purine on one side pairing with a pyrimidine, and same thing with A and T. If your teacher is into having you memorize like which is which, the acronym PAG, pure as gold, is helpful for remembering what which bases are pyrimidines. So I'll type that in the chat box because I don't have it written on my slide, but it's pure as gold, and that means purine, adenine, guanine. And pure as in like purine, right? Because you can't say, oh, they both start with P, but it's pure is the word, pure. So purine, adenine, and guanine are going to be your purines. Okay, nitrogenous base pairing. So here's a picture of them base pairing, G and C. And the phosphate sugar backbone on the top and the bottom. Okay, so you can see here the red dashed lines are showing hydrogen bonds between your A and your U and your G and your C. So this is a RNA molecule. And we know RNA is single-stranded, but this would be something where if you haven't gotten into DNA transcription yet, this would be kind of a snapshot of when DNA is being read and made into an RNA strand. Okay, so go ahead, take a look. Guanine and cytosine are capable of making how many hydrogen bonds? This is why they bond together. So go ahead and type the answer in. A1, B2, C3, or D4. So if you're looking at the diagram, we have G and C, we have A and U, but the question is asking about G and C. Why are they making that many bonds? Or how many bonds are they making? How many hydrogen bonds? Go ahead and type in your answer. All right, good job. Got one answer in. Good. Yeah, so your answer is going to be C, 3. It's making 3 hydrogen bonds. Again, that's kind of a stickler specific piece of information, but one of the skills that we want you to know for AP biology is being able to look at a diagram and make sense of it. And so you might be given just a diagram like this, and it says nothing else but, you know, what's being modeled here, right? It's obviously right in the middle here. The hydrogen bonding between the bases is what's being modeled for you. Okay, so back to our T-chart real quick. Added a few things. The bases found in DNA and RNA. So GCAT for DNA, GCAU for RNA, very good. And then hydrogen bonds between the bases for DNA, phosphate sugar backbone. I added this one in too, that they both play a role in genetics and heredity. If we would have had time, I would have gone over a little bit more of transcription and translation, but that is jumping ahead a little bit too much. So we'll wait until we're into energetics and we can talk about that a little bit more. Okay, so 5' to 3'. This is something that's going to be huge when you start talking about DNA replication. And so I drew this myself, and I recommend drawing diagrams, especially when it comes to molecules and really understanding on your own, making your own drawing of something, not necessarily a carbon copy of what's in your textbook, but making your own drawing, use color pencils, use markers or fun Sharpies or whatever. It really does help. So right now I've got 2 nucleotides joined together. Our bases are here, A and T. And I've numbered our carbons. So if you've taken AP chemistry, carbon numbering is probably very easy for you, and that's great. But if you're not, so we typically start to the right of the first oxygen. So this would be carbon 1, 2, 3, 4, 5. And then if you notice, this nucleotide is upside down. Okay, we'll explain what that means in just a second. All right, so if I were to add more nucleotides and more nucleotides like that, that's how they're going to be added. And so when we talk about 5 to 3', when DNA is being replicated, that means that the 5' end is being added to the 3' end. Okay, so I'll go back to where we started. Here's my original nucleotide, and so we're going to bring in a new one. It's the 5' end is being added to the 3'. And so when we talk about it going from 5' to 3', we're talking about which carbons are being interacted with. And the carbon numbering helps. Okay, and then the other thing to notice, the strand on the left is running, the very top is the 5' end, and look at the very bottom, it's 3. The bottom carbon is the 3, so we call that the 3' end. So we'd say this strand runs 5 to 3, and then the strand that it's connected to is running, look at the top. It has a 3 and a 5 at the bottom. So that one is running 3 to 5. Okay, so that's where that word anti-parallel comes from. So if you've heard your teacher say, oh, they run anti-parallel, if they ran parallel, both sides would have 5' at the top and 3' at the bottom. This one has 5 to 3 and the other one 3 to 5. So anti-parallel is how we describe DNA as well. And there's our other one added, okay? Here's just another picture of what 5' to 3' addition looks like. So I've got 2 nucleotides already. This one's being added to it. So the 5' end is being added to the 3'. So 5 to 3'. 5' end is added to the 3' end, okay? All right, so that's just another highlight. 5 and 3. So number 4, concept check. What type of bond is formed when the phosphate group attached to the deoxy or when the phosphate group attached to the deoxyribose sugar ring? So your phosphate and your sugar, what kind of bond are they making? One answer, A, hydrogen bond, B, ionic, C, covalent, or D, disulfide bond. So once we've connected those two ends, what type of bond is the phosphate sugar backbone using? Oh, I think 2 of you already answered. Awesome. Oh no, that's from the previous question. But all my answers are still the same. Wow. I keep using C as the answer. That's not what most teachers do. Sorry about that. It's a covalent bond. So this is a huge thing for everyone to know. The covalent bond is your phosphate sugar backbone. Okay. Here's another multiple choice question. So AP College Board's also really into models, and so they'll show you the same thing but 3 different ways, and you have to know why each model is the way that it is. So here's one model of DNA. It's not fully labeled, and that's on purpose. So 5 says, which feature of model 1 best illustrates how biological information is stored in a DNA molecule? So this gets at, okay, well, what does it mean to be biological information? How is it stored? Okay, so A says the dotted lines to represent hydrogen bonds between bases. So you have to ask yourself, okay, well, is it the hydrogen bonds between the bases that allows it to be a biological molecule? B says the 5' to 3' labels on each strand. Okay, so then you have to think, all right, well, does the 5' to 3' illustrate that it's biological, or is that just the chemistry and the organization? C says the lines connecting the sugars and phosphates to represent covalent bonds. Okay, so is it the covalent bonds that makes it a biological molecule? Or D, the linear sequence of the base pairs? So the actual sequence, A, G, C, T, T, A, C, G, 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 like that linear sequence, does that make it biological? Or biological information, I guess I should say. It's all biological. Great, yay, everyone's getting it. Maybe I gave it away with my tone. But yes, it's the linear sequence. That's what makes it biological. Okay, awesome. Okay, great job. Okay, 6. This is our last multiple choice question, and then we'll get to a free response, all right? And then we'll be all done. Number 6, which describes a structural similarity between RNA and DNA that's relevant to their function? So again, you might be able to have an awesome Venn diagram comparing and contrasting DNA and RNA, but when faced with a question like this, you might be like, oh shoot, this is super wordy. Where am I? And your head might start to spin. So we're going to look at each option at a time. So A says, both contain nitrogenous bases and phosphate groups, which allows each molecule to be used as a monomer in the synthesis of proteins and lipids. So looking at that, you can say, well, part of it's true. Yeah, they contain nitrogenous bases and phosphate groups. And it can be used as a monomer, right? But it says in the synthesis of proteins and lipids. Okay, so that is relying on, do you know what the monomer of proteins and lipids are, right? So our monomer for proteins is amino acids, and our monomer for lipids are your triglycerides or your glycerol and your fatty acids. And so I would be skeptical of that option. B says both are composed of the same 4 nucleotides, which allow each molecule to be produced from the same pool of available nucleotides. So we're looking at RNA and DNA. Are they composed of the same 4 nucleotides? No, thank you, Emily. No, they're not, right? That was one of the first things you guys said from the diagram. You said that one has U, this one has T, right? So that cannot be it. C, both are composed of the same type of 5-carbon sugar, which allows each molecule to act as a building block for the production of polysaccharides. Okay, so there's 2 things wrong with that answer option, right? They're not composed of the same 5-carbon sugar. That's why we have deoxyribose and ribose sugar. So that gets at partly, do you know what D and R stand for in DNA and RNA? And then also it's talking about for the production of polysaccharides, but from our carbohydrate information, we know that the monomer of a polysaccharide is a sugar molecule, not a nucleotide. Okay, so last one. Both contain nucleotides that can form base pairs with others, which allows each molecule to act as a template in the synthesis of nucleic acid molecules. So they both are a template. They can both base pair, absolutely. Okay, so even though if you were to just look at this question without it being broken up in A, B, C, D, I feel like at least my students in the past would get overwhelmed and they might just pick something that sounds right. Like they might have picked A, right? Because they're like, yeah, sounds good. And then they didn't read the part that says proteins and lipids. Okay, so when you get a big question like this, just take your time, read each part carefully. I would even annotate the question and cross out things that don't make sense and say, okay, well, it definitely can't be that one. And just, take your time on things. All right, great job. Okay, so here's your free response question practice. So we've got 3 models of DNA. I just drew these up. They're nothing fancy, obviously, but they're showing different things, showing different properties. If we were to even go back to that T chart of what's the difference between DNA and RNA and what's their function, each model is kind of highlighting a different thing. So for each model, describe which characteristics of DNA it best illustrate, explain the characteristic and how the model serves to represent it. And for something like this, don't be afraid to be wrong, right? So model 1, I would say there's probably 2 or 3 things that it's highlighting. So you could choose something, and if you have evidence to support it. So for example, like model 1, I know my picture is bad. I'll draw better next time. But this in the middle is showing 3 hydrogen bonds. This one's showing 2. This one's showing 3. This is showing 2. And the fact that it's 5 and 3 prime. I would, for model 1, talk about how it's showing that bases have hydrogen bonds. And that means that they can be easily broken, which means DNA can be opened up and used as a template for replicating. Or I would maybe even focus just on the 5 to 3 prime end idea and talk about why is it important that this model is showing that DNA is anti-parallel. So I would say the characteristic is that it's anti-parallel, and this is what anti-parallel means, and I would talk about it, right? And then say, yeah, so we've got this 5 prime end to 3 prime, and explain what you mean by 5 to 3, and how if they weren't anti-parallel, it would make replication really difficult. Also keep in mind, this is the first unit. This is only September, and so your ability to be able to explain stuff is not going to be as great as it's going to be in May when you take the test. In May, you're going to have all this information and all this experience to be able to tie in stuff from the evolution unit and the photosynthesis unit, and you're going to have information from a ton of different places that you'll be totally okay, right? Model 2, I would talk about the importance of complementary base pairing, right? This is really the only thing the model is showing, is that G and C fit together perfectly, A and T fit together perfectly, and I would expand on, well, why is that important? Why is complementary base pairing important? Okay, well, it's contributing to the genetic code, right? And so the sequence of those complementary base pairs is extremely important. And so if a mistake happens, it's catastrophic, right? Or could be catastrophic. Sometimes mistakes happen and it doesn't even, the cell doesn't even notice. Okay, model 3, last one. This is a really simple model, so I would even just talk about how model 3 is showing the characteristics of a double helix and why a double helix is important. It's a nice stable molecule. It's actually something I didn't talk about in the beginning, but DNA is a very stable molecule, this nice double helix. Talk about maybe the types of bonds the backbone is made up of, because you see I did this one strand in pink. So maybe that darker strand is highlighting the fact that it's like a strong backbone. And what is that backbone? It's phosphate sugar. So this might seem a little subjective in terms of, well, what should I answer? What's the right answer? It doesn't always necessarily, there's not one thing they're looking for. You can pull 3 or 4 different things from one model or question. And as long as you can explain it, with terminology and a good explanation, you should be in good shape. All right. Okay. We are done for tonight. Thank you so much for coming and joining us. Again, follow Think Fiveable and me as well. I'm on Twitter at Mrs. Parnin. Is there any questions? Usually like to leave a few minutes for questions. So if there's anything specific you have a question on, go ahead and type it in. Thanks, everybody. Great job. You're doing a great job by coming and watching these and interacting. It's going to help you a ton. Sunday will be my next stream. It'll be Unit 1 review. So we'll go over the whole entire unit, and then we'll start Unit 2 after that. All right. Thanks, everybody. Have a great night.